This is my Form 4 slide for enzyme. Some things from here we need it in our, the main concept is needed for our explanation. So I'll just explain here. So you do have immobilized enzyme in Form 4. But the explanation in Form 4 is we use the immobilized enzyme to overcome the problem of pH and temperature. Now you already learned if you make the temperature too high, the enzyme denature. Higher than optimum, the enzyme denature. pH, if it's not optimum pH, the enzyme doesn't function. So it's a limitation for enzyme. The enzyme is not perfect. It is sensitive to pH, it is sensitive to temperature. When it comes to industrial application, commercial, if a certain, and the factory is using, it needs to use certain chemical reactions catalyzed by enzyme, they are always thinking, how do we make this reaction faster? How can we finish the reaction in a shorter period of time so that we can reduce the cost? How to improve the reaction? So one of it is by using immobilized enzyme in Form 4. That's what we explained. By using immobilized enzyme, you can overcome the limitation of enzyme. Last time can only use optimum temperature. Now can use higher than optimum temperature because you have made it into immobilized enzyme. Last time can only use within certain pH. Now can use wider pH because you have made it into immobilized enzyme. So we use immobilized enzyme technology to overcome the problems in terms of factors affecting the enzyme. That is the Form 4 explanation. Under that, the example that I gave in Form 4, and this is one way of, this is all answers in your exam. How do you immobilize the enzyme? So we can attach the enzyme permanently to an inert substrate. Inert means they don't do the reaction. The support here is a substance that is not involved in the reaction. It's inert. They don't do reaction. We permanently attach the enzyme, maybe by using covalent bond, maybe by using hydrogen bond. The thing is, when the enzyme is permanently attached, they can't move. Mobile means can move. If cannot move, it's immobile. That's why we use the word immobilized enzyme. The enzyme cannot move. They are trapped. How they are trapped? Maybe they are permanently attached through covalent bond to an inert substrate. Now you might be wondering, why do we do this? Why should you make an enzyme immobile? Only when they are mobile, they can do reaction. They collide, effective collision. What's the benefit? The benefit is, because the question will come, advantage, disadvantage. The obvious disadvantage, the enzyme is not mobile. So less chance to have effective collision with the substrate. It's an answer. You are making it immobile, tak boleh bergerak. So kurang ke barang kalian perlanggaran berkesan, effective collision. That's the disadvantage. But the advantage, you can now use it at higher temperature. You can use it at wider pH range. You can reuse the enzyme. The enzyme doesn't contaminate the product. These two sentences. The enzyme doesn't contaminate the product. You can reuse the enzyme. How do we explain that? This is one method. Your syllabus, 3 methods to immobilize enzyme. This is one of them. I'm showing to my Form 4 students. Actually, Form 4 students no need this. Combined with inert substance, covalent bond. Or we can trap them in an insoluble capsule. Because it's insoluble, they are not involved in the reaction. You trap the enzyme, you see, this is entrapment. This is covalent bonding, this is entrapment. Entrapment means we trap them in a porous The word porous means the substrates can go in, the substrate can go in, the substrate can go out, but the enzyme cannot. Substrate can go in, product, substrate goes in, reaction happen, product comes out, but the enzyme is trapped inside. They are encapsulated or trapped in a porous, insoluble substance. This is for form 4. Later we'll make it more detailed. The thing is, the enzyme is trapped, the enzyme cannot move. What is the example? Okay, example, that some people have this problem, lactose intolerance. Lactose intolerance, meaning if they drink milk that contains lactose, they have allergy reaction. Some allergy reaction, they have diarrhea, vomiting, rashes, asthma attack. Colic means babies, they cry continuously. Supposed to be for babies, I think adults also do that generally. Colic means cry continuously. Now, if you had babies at home, you know, sometimes the baby will be crying and will be wondering why the baby is crying. Already give food, already give this, still the baby is crying. Sometimes we don't know. Maybe it's because of, like, there are many reasons. Could be lactose intolerance. So now, if the milk have lactose, some humans cannot drink it. Example, people who have lactose intolerance. So the factory is now thinking, okay, now how do I make milk that doesn't have lactose? Milk has lactose. When it's produced, the cow produces the milk, it has lactose. How to produce milk without lactose? So we use immobilized enzyme technology. So what we are going to do is this. This is the theory behind it. This is the example of milk, lactose-free milk, organic milk, half gallon, vitamin D. The cow is jumping, very happy cow. Organic milk mean what?
Organic.
Huh?
Natural.
Natural. I mean the cow is not natural. What? Then the cow.
Factory, yeah.
Then got inorganic milk also.
I think not produced in factory.
What milk is produced in factory?
I think milk that's not produced in factory or something like. What the name of the thing is?
I'm a bit curious why they mean organic milk. Okay, anyway, never mind. Not in the syllabus. They're not going to ask. Just my brain, overthinking. Because I know organic vegetable, we know lah, it's grown in without using pesticide. So that means this cow.
Not GM.
Oh, no, they didn't use any chemicals, I suppose. Anyway, never mind. Come back to syllabus. So now this milk has lactose. The milk, this bowl of milk has lactose. The theory is, or the what we can do is, the milk has lactose. So what we can do is we can add enzyme lactase inside. The milk has lactose, we add lactase inside. What the lactase will do, it will hydrolyze lactose into glucose and galactose. Lactase, we add lactase inside. So lactase will hydrolyze lactose into glucose and galactose. Problem solved. The milk doesn't have lactose. Now people can drink this milk because no more lactose. But we face another problem. The milk don't have lactose, but it has lactase, the enzyme. You put the enzyme inside what? If you don't remove the enzyme, the taste of the milk will be a bit different and people don't like. Now dilemma, how do you remove the lactase out? You solve one problem, you create a new problem. The original problem got lactose, now no lactose. But you replace it with lactase, now got lactase. Now how do you remove the lactase? This is the meaning of the enzyme contaminate the product. You put enzyme inside, they do a reaction. After that, how do we remove the enzyme? The enzyme contaminate the end product. It's in the end product. Hasil akhir ada enzim. Macam mana nak pisahkan tak tahu. Solution. We use immobilized enzyme. So what we do is now. So she's thinking how to solve this problem. This lady is quite famous. She.
Yes, I know.
I purposely put this picture. The Form 5 girls all won't like because the girls tend to overthink. Anyway, come back here. So now how do we solve this problem? This is a porous bead. That means the milk can pass through. It's a porous bead. Inside, we trap the enzyme. BEAD, bead, like guli. And it's permeable, but enzyme cannot go out. The enzyme is trapped inside. So we pour the milk. The enzyme is like this. We pour the milk. The milk have lactose. So the lactose will go in because it's porous. They go into the bead. The enzyme will catalyze the reaction. It will become glucose and galactose. And the glucose and galactose will come out. Now, how do you remove the enzyme from the milk? I'm asking you a question. How do we remove the enzyme from the milk? Now easy. How to remove the enzyme? The reaction done. So what to do?
Yes, I know.
How do you remove the enzyme from the milk?
If you use soy milk, there's no problem with this. Okay, stop.
Waiting more students to answer. So far only 2 students answer. How do you remove the enzyme from the milk? Waiting for answer from Jasmine T. Kong Xuan, you can't be using the aiding of the title line. Give me an answer. How do you remove the enzyme? Don't write the immobilized enzyme. Jasmine, so far Jasmine T and Anka Moon. I think so far 2 of you already got it correct. Now others.
Don't use milk. Use soy milk.
So why did we put the enzyme inside the bead? So that you can easily remove the enzyme. Remove the bead. When you remove the bead, because beads can be boleh ditapis, can be sieved. So you can remove the bead. So just take a sieve, pour the milk will come out, the beads will be trapped. The enzyme is not in the milk. So it makes it easy for us to remove the enzyme from the milk. The enzyme doesn't contaminate the end product. That is the importance of immobilized enzyme. So now if you see the product does not contain enzyme, product is not contaminated by enzyme. The actual technology looks something like this. This is the milk. You just pour it from the top. And this is commercial. This one, and we are showing you a small picture. It's like a huge tank. They pour the milk from the top. The milk will come down at the bottom. Commercially, it's a huge tank. Inside the tank, all of these green circles are the resin beads. RESIN, resin bead. They are not involved in the reaction. They are inert, but milk can pass through. Inside got enzymes, same like what I showed you earlier. So we pour the milk from the top. As the milk comes down at the bottom, they already become lactose free. This is immobilized enzyme. A short video showing you how it happens. So you inside here, this is the milk we already pour. This is a small scale of what happens in the actual factory. A model showing you what happens. We pour the milk, slowly the milk is flowing down. acid flows down, lactose will be hydrolyzed into glucose and galactose. You can see the milk slowly flowing down. But as the milk flowing down, take note of what remains inside the jar. What remains in the jar is the resin bits. Now, if I zoom in, you can see all of these are resin bits. Inside there is immobilized enzyme. The milk that came out at the bottom doesn't have enzyme. This is the meaning of immobilized enzyme. This is the Form 4 video. Now we have to come back to okay, enzyme application, no more. So now we have to come back to the reality of Form 6. Okay, now we come back here. Okay, so what I showed you earlier is this entrapment. Enzyme immobile, technology of binding enzyme to a solid medium so that the enzyme can be used and reused to carry out industrial process continuously. The video that I showed you, you can continuously pour milk. The enzyme can be reused. The main technique, entrapment. Physically trapped in medium such as silica gelatin or collagen. Okay, wait. Yeah. Entrapment means, as the name suggests, you trap the enzyme. There are two ways to trap. One way is what I showed you earlier, encapsulated in porous solid beads, which has selectively permeable membrane, such as calcium alginate beads. Now, the name calcium alginate, if you can remember, okay, write it. If not, just write up to here enough. Encapsulated in porous. The word porous means milk can go in, can go out. Any other substance involved in the reaction, which has selectively permeable membrane. If you can remember, right? If not, never mind. That is one way. Other way is you trap it in collagen fibers. That means the first one, entrapment. Okay, now this entrapment, there is two ways to do it. Now, the first way of entrapment is, this is the enzyme. Now, this is the porous bead. And inside, we put an enzyme. So, the enzyme is trapped inside or encapsulated. So, this one, we can also use the word encapsulated. So this is the first method under entrapment. It is encapsulated in porous beads. It basically means you trap it in beads that are porous, meaning substrate can go in, products can go out. Another method is we take this enzyme, we trap them in collagen fiber. For example, this is the collagen fiber. So the enzyme is trapped inside. Let's put a few. So the enzymes are trapped inside. This one also comes under entrapment. You are trapping the enzyme in a bead or maybe in a matrix of collagen fibers. Trapped in the fiber. That's the first one you have here. Trapped in a silica. The word lattice. Lattice means like a lot of fibers crossing one another. Lattice or collagen fibers. So here maybe we write, okay, silica lattice. Maybe we can write this. So this is the second type, silica or collagen lattice. Both comes under the word entrapment. Now the second method is what you saw in the picture earlier, covalent coupling. So covalent coupling means now we have a structure that is inert. So we'll use brown color. This is the inert structure. Now what we do is we attach the enzyme with covalent bonds. That's what you call as covalent coupling. So we just attach the enzyme by using covalent bond. This is covalent coupling. The second type. The third type is same, but just that now it's not covalent bond, it's weak bonds. So we call this adsorption. So all I have to do is I just remove these three straight lines and just change it into some broken line. So let's say so the broken line is to show you it's not like hydrogen bond. So it's not a strong bond, but it keeps the enzyme in place. So this is the last one. We call this adsorption. Lekat. This is adsorption. So this is the three different methods of covalent coupling, adsorption, these are different ways of trapping the enzyme. Or not trapping, immobilizing the enzyme. The definitions are in here. You can. So adsorption is adsorbed by weak bonds. Covalent coupling is through covalent bond. Entrapment is you trap in a bit or maybe trapped in a network of fibers such as silica or collagen fibers. I've drawn the picture. Hopefully it's easy for you to understand. Advantages, disadvantages. As I mentioned, you are immobilizing the enzyme. So less chance for it to collide with the substrate. Rate of reaction may be lower. Active site may be covered by inert matrix, or there may be less chance for the substrate to attach to active site. It might be expensive to produce immobilized enzyme. Advantages. End product are not contaminated by enzymes. You don't have to purify the end products because they don't have enzyme. They are not contaminated by enzyme. The enzyme can be reused, so it reduces the cost. They are more stable, can be used at higher temperature, larger pH range, can withstand denaturation. Semua ini tadi saya dah terangkan. For some reason, immobilized enzyme is famous. Many times the question came up, immobilized enzyme. Banyak kali soalan keluar. I would say in the new syllabus, among all the enzyme question, we are at the last question now. You learn so many enzyme question. Immobilized enzyme is famous. Banyak kali keluar. And if it comes out, they will ask you advantage, disadvantage. So here one question, 2050. Resin beads are used as matrix in entrapment technique of enzyme immobilization technology. State the roles of the resin bead and advantages of enzyme immobilization. I already show you there is four advantages here. How do you do entrapment? Why you do entrapment? These 5 marks is considered bonus. You write back the advantages and you write back what is entrapment. That should be enough. And preferably you add in the definition of enzyme immobilization. Famous question. Before I go to the next page is still enzyme immobilization example. Before I go to that, any question about this page? The page of enzyme immobilization. Any question? Let me lower your hand. So kelas hari ini bukan susah sangat. Previous classes were quite complicated. Today's class is quite direct. Not as complicated as enzyme kinetics, Michaelis-Menten inhibitors. Quickly we can finish it off. So we are already at the second or third question. We are at the third question. Normally one day I do only one question. We are now already at the third question already, and we are finishing the chapter. So now you already know what is enzyme immobilization. Example. So there's two examples that you need to know. Okay, so application of enzyme application is something called as biosensor. Now the word bio basically is biological material. Okay, in the word biosensor, the word bio is a biological material. This biological material could be hormone, could be enzyme, anything that is produced by living organism. The sensor, the meaning of the word sensor is they can detect. That's the function of sensor. They detect something. So, red color. Okay, this one is detect chemicals. So using that word, so what is a biosensor? It is a device that can detect chemicals accurately and quickly, but it uses biological material. A device that uses biological materials to detect chemicals quickly and also accurately, from the word biosensor. So it's a probe that integrates a biological material. The word integrate means is part of it. Part of the device is a certain substance produced by living organism. It integrates a biological material with an electronic component to detect specific chemicals accurately and also rapidly. Actually, bio is biological material. Detect chemical is the word sensor. Biosensor is an electronic device. When you combine both words, could you try again? Siri responding to me. Okay. So this one is electrical device. That means the Siri is the phone is listening to everything that I'm explaining. So that's how Apple, Google, all of these companies knows what you're doing. What you're talking, not only you and your friend are talking, your handphone is also listening. A clear example is the Siri asked me a question, what you're asking. So your secrets are not with you. The handphone company, Google, knows all your secrets. Okay, anyway, come back here.
You mean Apple?
Apple and Google, mainly Google. Google listens to everything you're saying.
Yeah, I thought, but Apple made Siri.
Yeah, Apple, Google, everything.
No.
Your handheld device, all listen, all got ears. Okay, anyway, biosensor is electrical device. Bio is biological material, detect chemicals is the sensor. So you combine all of that, you have the definition of biosensor. An electrical or electronic device or electrical, should I use electrical or electronic? Sorry, we'll change this electronic device that detects chemical quickly and precisely, and it integrates biological material. And normally, because enzymes are precise action. If they don't attach complementary, the reaction won't happen. Hormones are also specific action. So biological materials are precise. So if you want to detect certain chemicals, using biological material makes it even more accurate and also fast. So, but because you are now using enzyme, for example, you are trapping it in the device, it comes under immobilized enzyme. We trap the enzyme to detect certain chemicals accurately, precisely, but it's not limited to enzyme. Hormones also can use. So there is two types of biosensor. Now the example we have in the that we use is how to detect blood glucose level. So there are two ways to do it. One uses electronic component, another one doesn't use electronic component, meaning one uses electricity, one more doesn't use.
I did it.
Okay. So one uses electricity, one more doesn't use. The one using electricity is, okay, this one using electronic. The one that uses electronic measurement. Or maybe we first do the one that doesn't use electricity. So now this is a dipstick. So let's say this is the name is dipstick. Something like an ice cream stick. The shape is almost the same. So now this is a dipstick. The name dipstick because you dip it into the blood sample or the urine sample. You dip it, a stick that you dip into the sample. That's why you call it dipstick. So this is the dipstick. Okay, now normally the dipstick is made of cellulose. So it is made of cellulose. So this is a cellulose strip. It's made of cellulose, and inside the cellulose strip there is immobilized enzyme. So there is immobilized So there is an enzyme by the name of glucose oxidase. So far you haven't used it. So there is immobilized glucose oxidase, and there is also immobilized peroxidase. So inside the dipstick already had immobilized enzyme. There is also one more, which we call as colorless hydrogen donor. Now, to represent this colorless hydrogen donor, you can use any alphabet. So I'm just using AH2. The A is the donor. They at the moment they have 2 hydrogen. Later they can donate the 2 hydrogen. We'll explain. So colorless hydrogen donor. So all of this is inside something called as a dipstick. This is a dipstick. It's a cellulose strip. It contains immobilized glucose oxidase, immobilized peroxidase, and colorless hydrogen donor AH2. You take this dipstick, you place it inside a blood sample, and once we dip it inside the blood sample, what happens? The first reaction that happens is this. Okay, I didn't write the formula. Okay, here. So dipstick consists of glucose oxidase enzyme, peroxidase enzyme, and the hydrogen donor immobilized into a strip of cellulose. I already draw for you. When you dip the dipstick into a blood sample, glucose oxidase in the biosensor oxidize glucose into gluconic acid and hydrogen peroxide. I want to write the formula. So the enzyme that is going to do this reaction is glucose oxidase. So the name of the enzyme is glucose oxidase. So the immobilized glucose oxidase will catalyze oxidation of glucose to form gluconic acid and hydrogen peroxide. Now the next enzyme, which is immobilized peroxidase. Immobilized peroxidase will now catalyze the reaction between hydrogen peroxide and the colorless hydrogen donor. Now take note, all of this is only possible if glucose is present. If glucose, let's say we are doing this in a urine test, if the glucose in the urine don't have, then the whole reaction won't happen. If glucose is present, and also the amount of glucose, the more glucose present, the more gluconic acid can be produced, the more color change will happen. So not only it depends on whether glucose is present, it also depends on how much glucose. The more glucose present, the more color change will happen because of this colorless hydrogen donor. The second reaction, I already have it here. So the colorless hydrogen donor reacts with hydrogen peroxide. We get water and A. A is a colored substance. When they donate the hydrogen, they become colored substance. So the dipstick color will change. So maybe let's say this part, the color will change. Maybe here turns red, for example. After you dip it into urine or blood, after the reaction, the color will change. So based on the color, based on the intensity, you may have blue color. It might be dark blue or light blue. So the color and the intensity, we compare to a scale, same like your pH paper, same like your pH paper. You compare it with something like this. So you can see the dipstick here. Okay, maybe the picture not clear, but you have the idea. So after you do the test, let's assume this is the dipstick. So you compare it with a scale, like you see the green color itself got a yellow color, got many, the green color got many. Based on the color, based on the intensity, you can identify whether there is glucose present, and also if glucose is present, what is the concentration. So that's what you have right at the bottom here. Peroxidase catalyze reaction between hydrogen peroxide and colorless hydrogen donor to produce a colored compound. So the dipstick color will change. The type and intensity of color when compared to a scale indicates the concentration of glucose in the sample. It can be a blood sample, it can be a urine sample also. This one, no need electricity. Dipstick doesn't need electricity. So if you're going to outskirt pedalaman, no electric supply, then this is convenient to use. But in a laboratory setting, you want even more precise measurement, then we can use biosensor that uses electronic device. The difference will be the first reaction is same. This reaction is same. The first reaction is same. We need to detect the concentration of hydrogen ion. Let's see what we have for a biosensor that uses electronic device. So the first one, the one we explained doesn't use electricity. Now this one. Glucose oxidase in the biosensor oxidize glucose into gluconic acid and hydrogen peroxide. Now gluconic acid is the acid. So the acid releases hydrogen ion. The sensor will detect hydrogen ion. The concentration of hydrogen ion will be detected, converted into an electrical signal. So the acid or the hydrogen ion produced are measured and used to produce a measurable electrical signal. Now first, the change in pH is converted by a transducer into an electrical signal. Up to here, 1 mark. I combine the sentence, but if biosensor comes out, the change in pH is converted by a transducer. The name transducer is needed. Transducer changes a chemical signal into an electrical signal. The pH is chemical signal. Changing into electrical signal is the function of transducer. But the signal that is produced is normally very weak. So if it's too weak, sometimes you don't get a significant reading. So to make sure small change also you can get a larger reading, we have to make the signal stronger. That is the function of the amplifier. So the signal is then amplified by an amplifier is another 1 mark. Transducer is 1 mark. Amplifier is another 1 mark. Transducer changes chemical signal into electrical signal. I wrote all in one sentence. You make it into 3. The electrical signal is amplified by using an amplifier. Now that signal needs to be converted into something that is readable. Maybe some chart or maybe some wave, maybe some numbers. That is the function of the next one. And is finally displayed as an electrical or optical signal. The amplified signal is now displayed as an electronic or an optical. Optical means something you can see. Optical signal. You write back the same thing. Define what is biosensor. Mention the first reaction. Mention what they detect. Mention what changes chemical into electronic. What amplifies the end result. So 1, 2, 3, 4, 5, 6. Let's say 5 marks. Always when biosensor comes out, or always when biosensor comes out, enzyme immobilization comes out, they will ask you advantage, disadvantage. Disadvantage, expensive. Maybe to biosensor to create expensive, for example. Maybe the disadvantage we already write here. Earlier we already write the disadvantage. But this is advantage, disadvantage of immobilized enzyme. Biosensors have their own answer in the next page. Advantage, disadvantage, down here. Accurate, quick, advantage. Very sensitive, can detect low concentration of chemicals, advantage. Safe to use, as cost can be reduced if mass produced. Mass means you produce in large quantity. All of these are the advantages. Or even this one also. Helps in early detection of medical cases. Treatment can begin early. I don't think I have this advantage. Yeah. These are all advantages. You can also add all of these as the answer. Advantages. You can choose some from here as the advantage. You can use the disadvantage from, because this is also immobilized enzyme. Biosensor is application of immobilized enzyme. So you can write all of this as the advantage. You can also write These two, whichever you feel necessary, you can put it in the answer. If you feel your answer not enough, you can add all of this inside in the explanation. Not all, you have to choose which one can be used and add it into the answer. Because this is also immobilized enzyme. There should be a question about immobilized enzyme. Recently, and it's recently, okay, maybe next class I'll show you that there should be a recent question about biosensor. There was one question. Okay, anyway, we are done with biosensor. There is two explanation. You can, the one that is longer answer is the one that doesn't use electricity, this answer. But using electricity, you need to make sure you use the precise words, transducer, amplifier, and then the optical signal. That make sure that is worth 3 marks. And many students, normally these 3 sentences, they don't remember. So we are done with enzyme. It's a long journey for enzyme. All kinds of explanation was given for enzyme. Any question for this biosensor? Any question for biosensor? Okay, so the good news is we are done with chapter 4. So we finished chapter 4. So chapter 3, 1, 3, 4 done. 5 and 6 are heavy chapters. And since we have only 1.5 months, I think we have to do maybe this month 3 weeks. We might do 3